Thanks, Cheyenne. This talk is about some recent work. I've been having more fun than I ever had in my life, even more than I had when Cheyenne was at MIT. First of all, happy birthday to Ingrid again. She provided a wonderful excuse for a wonderful workshop, and very special thanks to Rebecca Willett. A poll among the organizers said she organized everything. So, thank you. Next week, I believe NSF will announce the outcome of the last competition for the Science and Technology Centers. And it looks like we're going to get a center for brains, minds, and machines. Its rationale is, I'm mentioning because it's a message also for the community here, I hope, looking at the future, is because if we want to make progress in the science and the technology of intelligence, I think we'll have to combine not only machine learning and computer science, but also recent progress in cognitive science and neuroscience. That's essentially, supporting what Cheyenne was saying about what I was going to try to do, to convince you that that's important. And the center is mainly MIT and Harvard, with a number of other institutions, combining computer scientists like Leslie Kelbling and Patrick Winston and Leslie Valiant, with neurosciences like Nancy Kanwisher and Bob DeSimone and so on. And also including a number of companies that have provided some of the recent progress in intelligent technologies, like especially Mobileye and Orcam in Jerusalem. Now, okay, so this is the background to tell you why I've been looking at the brain. But if you look at what I'm going to say from the point of view of machine learning, I think it's in trying to go beyond the first phase of machine learning, which has been quite successful for the last 20 years, and which is essentially based on the approach of getting as many supervised, labeled examples as possible. Is n going to infinity, is essentially the Google approach. Or what we did, for instance, 15 or 20 years ago to develop a pedestrian detector system was based on many examples. At the time, there were thousands. Now, Mobileye is using millions of labeled images for training a classifier. Now, a reasonable bet is that the next phase of machine learning will be using supervised learning, but a pre-processing stage based on unsupervised learning of good representations for supervised learning, so that the machine can learn with very few labeled examples, like children seems to do. So let me go into this. That's exactly the topic here. The question came looking at visual cortex made up of several areas, from the retina to V1 to V2, V4, and the so-called infratemporal cortex. And there has been models based on what we know about the circuitry and the connectivity and the properties of neurons in these areas. This is a circuit diagram of part of visual cortex. And one model we developed in our group, but was one of several models by other people, like Fukushima especially, and based on ideas of Hubel and Wiesel, is a system which is multi-layer, has at the bottom simple cells which are Gabor-like filters, and then complex cells, C1, that essentially pull together the output of these filters, these simple cells, with the same selectivity, which means orientation and wavelength of the wavelets, to get some invariance to translation and scale, and then iterating this in higher areas. And the surprising thing about this model for me was not that he reproduced properties of neurons in various areas, predicted a few of them, but also that he did pre-processing for recognition with a classifier at the top, which years ago was in par or better than the best computer vision systems. So just by imitating cortex, this was not what we were planning to do, it was a surprise. Now, so the question is, why do systems like that work so well? And the convolutional networks, nowadays called deep learning networks, are also, I would say, unreasonably successful in some of the recent tests in vision and speech, and there is no theory of why they do as well as they do. So, and this is in vision and speech. So this been working for the last 18 months or so towards a theory, and it's still unfinished, is not been published, but I want to tell you a few things. It seems to predict, from assuming that invariance is the main goal of this pre-processing, it seems to predict properties of cells at various levels of cortex. It may explain why these deep convolutional architectures, or HMACs, are doing as well as they do, and it's from a more global point, can become a theory of unsupervised learning, of how to learn good representations for supervised learning. And good representation for supervised learning means representations that reduce the sample complexity of the classifier at the top. These are the people involved. Fabio is a postdoc, and Lorenzo is visitor faculty from Italy, and then students. Now, the main assumption is that the computational goal of the ventral stream of a part of visual cortex involved in object recognition may be to compute a representation. Think of it as a feature vector, which is invariant to transformations observed during development of the organism. And here is a motivation for it. Suppose I do the following experiment. I show an image of one dog and one horse to a classifier, and then I test the classifier on other images of dogs and horses. And if I do this, the performance is, we've done the experiment, the classifier is a simple classifier on pixels. Not surprisingly, performance is chance. And even if you increase the number of labeled examples that you give to 3 pairs or 6 pairs or so, performance does not improve. However, if you assume that you have an oracle that gives you, that essentially normalizes the pose, the appearance of these images, so that the cats, the dogs and the horses are always from the same viewpoint, the same distance, the same position, then you try with one example of one of these dogs, one of these horses, and immediately when you test on all other images, you find performance that is 70%. That's a linear classifier on pixel, the red line. The blue one is a linear classifier on Gabor filtered images. So that's the motivation, is intuition that the sample complexity becomes much better. And there you can derive learning bounds in this, for instance, using ideas about compression that you effectively get by normalizing for these nuisance parameters like position and scale and viewpoint. Okay, so that's the motivation. Let me define the transformation I'm dealing with, mainly as geometric transformation, so the transformation of the coordinates. I'm especially concerned, initially anyway, with a affine group in R2, so is the general linear group in 2 dimensions plus translations. And then let's make a few other assumptions that from the point of view of neurons, that images don't need to be represented, are very likely not represented in cortex as pixels, but in terms of measurements on them. So you can think of them as as functionals. I would say linear functionals, mainly because it turns out that if you think about what is easy for neurons to compute, that's high-dimensional dot products. For a single neuron has in the order of hundreds to 10,000 synapses. You can think of each one as essentially performing a product between the weight of the synapse and the input there. And so then having the soma, the axon, summating all this multiplication, so a dot product. Okay, so the next step is to think about what happens when I transform an image. So for instance, I rotate an image. That's a compact group, and I can associate an orbit to this group in the usual way. So the orbit is the set of images generated by a starting image by the action of the group. Depending on whether the group is finite or not, I can have finite number of elements in the orbit or an infinite number. The interesting thing is that if the orbit is an interesting quantity because it's invariant, contains all the transformations, and it's unique because if 2 orbits touch, intersect, then they are the same. Okay, so how can I measure an orbit? We can prove that I can associate a probability density to an orbit, and knowing that is equivalent, perfectly equivalent to the orbit, is probability density over the group. So you think of this multidimensional histogram of probability of pixel x1 being so, x2, and so very high dimensional, taken over all the images in the orbit. Then there is a set of theorems, Kramer-Wald theorem, suggesting that you can get a proxy for this unwieldy high-dimensional probability distribution or histogram in terms of projections on a set of images or templates. So instead of high-dimensional probability distribution, you have now a one-dimensional, several one-dimensional probability distribution indexed by K. And we have some results I'll show you in a second about bounds on K to get an approximation of this probability distribution. Then the key observation from the point of view of neuroscience and computer vision is the last one. It's mathematically trivial, but is the fact that in order to have invariance for an image I, say the image of Cheyenne face, I don't need to observe all the transformation of I, especially from this point of view if I use projections, because what I can do is to look at the projection of I on all the transformation of a template. So suppose that when I was in the crib, a baby, I saw suppose, the face of my mother or some objects rotating, and I recorded a movie of this. So a sequence of images. This would be the GTK, and I store them for later use. And when I want to get an invariant representation of one single image I've seen of Cheyenne, what I do, I compute these dot products of this single image I with all these frames in the movie, and I take an histogram of it. And the claim is that histogram is invariant. In other words, if I compute it again from an image of Cheyenne that was now rotated, the histogram would be the same. So I don't want to bore you with, I can give you reference if anybody's interested, but can have a norm between, a distance between probability distribution and a result which is a bound on the number of projections that you need in terms of to approximate sufficiently the original probability distributions. And these results depends on the number of orbits or images you want to discriminate and not on their dimensionality. It's a little bit in the spirit of Johnson-Lindenstrauss, but it's completely different underlying mathematics. So we have this series of connection between equivalent images if they have the same orbits. The orbits are equivalent to a probability distribution associated with them, and then I can use these projections of this high-dimensional probability distribution on a set of templates, and because of this property of the group, I don't need to observe the orbits of the image I want to obtain an invariant description, but I need the orbits of the templates. Okay, so the way to compute invariance for neurons would be to have a set of simple cells that are basically doing, say, vertically oriented simple cells are doing computing dot products of the image, which the template they represent, transformed, for instance, in position, and then complex cells would pool the output of simple cells and compute histograms or some other quantity associated with it, like, for instance, the first moment, the average, or the second moment would be an energy model of complex cells. They are all invariant quantities, and with enough templates, they are epsilon unique in terms of representing the image. This is pseudocode for it, and here are some examples. For instance, suppose I consider just rotation, that's the simplest case of a compact group, and I have on the right two different faces. So when I compute the dot products of each one of them with the movie of this template face being rotated, I get an histogram which is different for each one of them because they are two different faces. But if you see here the two faces are one at the top, one at the bottom, and look at the top, independent of the angle of rotation of the face, I get the same red histogram. At the bottom, independent of the rotation of the face, I get the blue histogram, and they are invariant to rotation and discriminative, different from each other. Okay, so see, I have probably 5 minutes. Let me go. What I said is just a simple case of compact group, but we have extended this to non-compact groups or groups observed through a receptive field, a finite window, to having hierarchies of these modules, and also to non-group transformations. Let me try to mention very briefly here. For non-compact groups, this would be the case of translations. The interesting point that seems to emerge is that in order to have invariance, you need this dot product between an image and a template to be zero outside a certain range of a transformation. This is a symmetric difference on the right. Let me show you a simpler examples here. Suppose I have a, for instance, a translation group, and I have a pooling region between minus b and plus b. Then I have to have that the dot product between the image and the template is zero for transformation of templates, shifts in this case, that are larger than a certain quantity that must be smaller than the pooling window. Otherwise, it's essentially to avoid border effect in a sense, but what is interesting is, is that this is essentially a property of localization of the template or sparsity of the template in set of images. The condition is there that this dot product between I and the transformation of T is zero for this R, the transformation parameter larger than A. Then I have invariance within the range above. And so it seems that localization implies invariance, and probably that's a question that is a bit open. I have an answer in a simple case, is that invariance implies localization. And it seems that if you want to have optimal localization in optimal invariance for scale and position, you are bound to get something like Gabor wavelets, because they will become essentially constrained by this localization property to a Heisenberg box in frequency space. There is a similar sparsity condition when you assume that you want to have this localization condition not for all images with a certain support, but for only a certain class of images. I'll come to that in a second. So the extension to hierarchies is essentially to repeat the same idea of these modules of simple and complex cells, of doing dot products and histograms on them from the first layer to a second layer by considering the output of the first layer as an image that would be input to the next layer. So the diagram is like this, starting from the bottom where you have a small receptive field doing this operation. This would be like a complex cells looking at the world through this window that becomes larger as you go higher up. And we have results showing invariance, stability, uniqueness in these multi-layer structures. The main reason to have this multi-layer organization is because you don't want to have invariance or a signature which is invariant for the whole image. I don't want necessarily to see this whole scene and being able to recognize in invariant way. I want to be able to recognize in invariant way parts of it and parts of parts. So the idea of having invariant signature over different patches of the images at different stages of the hierarchies. There are other reasons, like minimizing clutter effects. These are the four main reasons for the multi-layer architectures. And then for the non-group transformations, a typical example I want to show very briefly is what happens if instead of a fine transformation on the plane, I consider transformations that are not groups, like the transformation induced by rotation in depth of a face. okay? That's a group in 3 dimensions, but not in 2. And another one would be change of expression of a face. I'd like to recognize a face invariant to expression. But let's consider rotation in depth. And you can do the same thing I mentioned, so this computation of dot products and pooling histograms on the output of Hmax. So you have already invariance to position and scale to some extent in Hmax, and now you want invariance to rotation in depth, and you can achieve it if the templates you use are templates of an exemplar of the same class, so other faces. So that the difference, before any template was fine for translation or rotation, here I need templates that transform in a similar way. And if I do that, what happens is that I don't get perfect invariance. This is recognizing a face independent of rotation in depth, starting from a frontal image. I don't get perfect invariance, that's the blue line, but I get much better invariance than without using this layer of face-specific invariance. Notice that this also kind of predicts the existence of modules in the brain for different classes. And we know that there are face modules in the brain and body modules and so on. We tested this recently on thousands of images, one of the faces, labeled Faces the Wild database. And the short story is that our technique, the blue line seems to do much better than other ones. This is an example of using Hogg histogram of gradients without and with this stage of pooling of templates of faces, dot products and pooling. So let me, I don't have time to go through another part of the theory that essentially predicts Gabor-like tuning of simple cells in V1 based on Hebbian plasticity, and also predicts some funny neurons that are tuned to one face view and another symmetric one in one of these face patches in the face area. This comes from solving an eigenvalue problems. Anyway, that's, let me skip this, but let me just finish saying that I hope this is a first step towards this goal of learning in an unsupervised way, invariant representation that make supervised learning possible. And the key here is invariance. I'm a physicist. In physics, you can essentially write the old classical physics and most of quantum mechanics from a small number of symmetry or invariance properties or conservation properties, and it would be kind of a dream if some of a similar invariance approach would be able to predict properties of cortex. Thank you. Right.
So, but yet, so I guess in some sense, Jan and Jeff Hinton are not up to your theory of work. Is that?
That's correct. That's correct. In fact, the system, which is a convolutional network like Hmax is, like visual cortex is, at least to the level of V1, the system that did very well on ImageNet, very well, much better than any other system before, was trained with labeled examples that were more than 1 million or so. I don't think a human baby gets so many labeled examples in his first 3 or 4 years of life, or ever. So I think the convolutional part, which gives invariance, hardwired invariance to translation in XY, helps already a lot, but not enough to reduce the sample complexity. But you can do better, that's my claim. Yeah, this is, I don't think, and I do not have time to say that this explains the whole of vision. This explains, at best, the first 100 milliseconds, the time it takes from the signal to go from the retina to infratemporal cortex. There are then back projection, top-down effect, moving your eyes, and so on. So there is a lot more going on if you wait longer than 100 milliseconds. So the first answer is that I think these other things, that you're right, are part of the rest of intelligence, which is probably much larger than this. But it's good to start from somewhere.